Respiratory system. Main questions. A respiratory system consists of two parts: airways and respiratory division. Airways consist of nasal cavity, nasopharynx, larynx, trachea, and bronchi, and ended by terminal bronchioles. After that, the respiratory division. Functions. Airways provide the chemical analysis and conditioning of inspired air, its purification, moisten, heating, and respiratory division provides a gas exchange between the air and blood, so supply blood with oxygen and remove carbon dioxide. All organs of airways have a similar pattern of the structure. They are tubular. They all consist of three membranes: inner mucous membrane, middle fibrocartilage, and outer adventitia. Mucous membrane consists of four layers: pseudostratified ciliated columnar epithelium, the first layer, surface epithelium, underlying loose connective tissue, lamina propria, then lamina muscularis, not developed in the upper parts of airways, and submucosa, loose connective tissue where the mixed glands, secretory portions located. Fibrocartilage consists of connective tissue and hyaline cartilage. You see the zone of mature cartilage with isogenic groups, the zone of young cartilage with chondrocytes, young chondrocytes, and perichondrium, where the chondroblasts and stem cells located. And adventitia come in these inspire air to airways. First, nasal cavity consists of vestibular and respiratory as well olfactory regions. Vestibular part lining by thin skin with hairs and glands. Respiratory part lining by mucous membrane, this simple columnar ciliated epithelium. In lamina propria and epithelium there are a lot of blood vessels heating the inspire air. And olfactory region lining by olfactory epithelium, this is a organ of olfaction. Then nasal pharynx, it's tubular organ. This is a way of air from nasal cavity, nasal pharynx, through the oral pharynx to larynx when the epiglottis is opened. Larynx wall consists of mucous membrane, fibrocartilage, and adventitia. It's lining mostly by typical respiratory epithelium, simple stratified columnar ciliated epithelium, for exception of vocal folds and epiglossus. It lining by stratified squamous non-keratinized epithelium. From larynx, the air enters the trachea. It's a tube contain between 60 and 20 semirines from hyaline cartilage. The ends of semirines connected to this connective tissue and smooth muscle tissue. It's because posteriorly behind the trachea there is an esophagus, and it's necessary for easy food entering through the esophagus. The cartilage of trachea could not prevent the entrance of food. And then air enters the bronchi, main bronchi, principal bronchi, and large and smaller bronchi, bronchial tree. This is the structure of trachea wall. The third and fourth layer not pronounced. Lungs are parenchymal organs covered by serous membrane. It's visceral pleura. It's lucid, and you can see the lobules, connective tissue septum dividing this organ for lobules. And inside lungs there is a continuation of airways, the bronchial tree. When the main principal bronchi divided for lobal, segmental bronchi, for large, middle, small bronchi, terminated by terminal bronchioles. Size, diameter of bronchi significantly decreased. Bronchi 16 times divided for 2 branches, and from one principal bronchi appeared more than 1,000 small bronchi. So it really looks like bronchial tree. And after that the respiratory portion started. It consists of respiratory bronchiole, alveolar ducts, and alveolar sacs, and everywhere are vesicles, semivesicles, alveoles. Only in alveoles the gas exchange between blood and inspire air take place. There are significant changes in the structure of bronchi wall, from main to small. This picture demonstrates these differences. All bronchi are tubular organ, consist of 3 membranes. For example, in large bronchi there are mucous membrane, fibrocartilage membrane, and adventitia. The mucous membrane consists of surface epithelium, stratified columnar ciliated epithelium. Lamina propria consists of loose connective tissue, lamina muscularis. Submucosa contain a lot of mixed glands. In large bronchi, the fibrocartilage layer consists of large islets or islands of hyaline cartilage. Previously, in principle, in main bronchi, there were full rings of cartilage. In middle bronchi, only small islets of cartilage. the middle membrane, and few glands in submucosa. In small bronchi, no cartilage, no glands, but lamina muscularis developed quite good. So the small bronchi can contract and relax and regulate the entrance of the air into respiratory portion. Terminal bronchioles lining by simple cuboidal epithelium, then respiratory portion consists of respiratory bronchioles, alveolar ducts and sacs, and everywhere the increased amount of alveoles. Near the bronchi you can find quite big blood vessels, arteries and veins. There are many types of cells in airways epithelium. Main cells are ciliated epithelial cells. They contain 200-300 of cilia in their apical portions, and they can move. Under scan electron microscope they looks like grass in the field. The cilia can move and remove the dust from the air. Goblet cells, like in intestine, secrete mucus, which cover the surface of epithelium and absorb the dust. And then cilia remove these mucus droplets with dust. Inserted cells or supporting cells between some endocrine cells. Individual endocrine cells belong to diffuse part of endocrine system. Basal cells, stem cells, are responsible for regeneration. There are also some dendritic cells of Langenhans, intraepithelial macrophages, antigen representing cells, like in epidermis of skin. In terminal and respiratory bronchioles, the above-mentioned cells are replaced for secretory Clara cells. They secrete, inactivate toxins in inspire air. There are also non-ciliated cells with microfolds, antigen represented cells, like in small intestine under lymphoid follicles. Hema-sensitive cells with brush border contain microvilli with chemoreceptors for chemical analysis of inspire air before it enter the respiratory division for gas exchange. A respiratory division consists of 150,000 of acini. Acinus is functional and structural unit of respiratory division. It consists of respiratory bronchial alveolar ducts and sacs. It contained up to 2,000 alveoles where the gas exchange take place. So every alveoles surrounded by blood capillaries. Two sources of blood supply through the bronchial artery provide the nutrients and oxygen to lungs, and another belong to small cycle of blood circulation. Pulmonary artery brings the venous blood, contain carbon dioxide from right ventricle of heart, and after the gas exchange it's saturated by oxygen and release carbon dioxide and come back to the heart through the lungs vein, pulmonary veins. And these yellow vessels, lymphatic capillary and vessels which collect lymph from the lungs. This is a acinus with alveoles. Its alveoles are semivesicles, lining by flat epithelium, squamous epithelium, and surrounded by blood capillaries. These black lines are elastic fibers. This is a key figure which explains the structure and function of alveoles. This is one alveole. This blue air, it's lining mostly by flat cells, type 1 alveolar cells. This is a blood capillary with blood, erythrocytes. It's lining by endothelium and very thin cytoplasm without nuclei of endothelial cell, and similar thin cytoplasm without nuclei of alveolar cells, a common basal membrane. These three structures together compose the barrier between air and blood, air-blood barrier. It's very thin, up to 0.5 micron, because 0.2 thickness of alveolar cell, 0.2 thickness of endothelial cells, and 0.1 micron the thickness of basal membrane, so 0.5 in general. Summary square of alveoles more than 100 square meters. It provides the best conditions for the quick gas exchange between blood and alveolar air. For few seconds the oxygen from inspired air diffuse into the blood and connected this hemoglobin and carbon dioxide are released from blood into the air. The second type of alveolar cells are type 2 alveolocytes. These are secretory cells which produce surfactant. This dark blue line which lining all alveoles. They consist mostly in phospholipids, lining the alveoles from inside and prevent the collapse during the expiration, and also prevent release of blood plasma from blood into the lumen of alveoles, and prevent the entrance of bacteria from air into the blood. There are type 3 alveolar cells, macrophages, alveolar macrophages. They phagocyte bacteria and particles of dust from the air. Smokers, or in minus, on this section, they provide the black color to the lungs. This slide demonstrates these secretory cells which produce and secrete surfactant, and this electron microscopic picture demonstrates the structure of surfactant. It consists of liquid and membranous phase. This is type 1 alveolocyte, basal membrane and endothelial cell, so this is a barrier between air and blood. So the membranous phase consists of several universal biological membranes in structure. So phospholipids bilayers these proteins.